clapping to come. Okay, so I'm excited to share some of the work that's in my dissertation with you guys today. Okay, so individuals often incorporate information about the social environment into their decision making. And this can result in social environment dependent behavior that can have fitness implications, especially in the evaluation of conspecifics, as is often apparent in studies of mate choice and male competition. So for individuals living in dynamic social environments, we would predict that they should adjust their behavior to appropriately match the social context that they face. So before we can really go into this work, then one question is, what social signals are individuals cueing in on to evaluate the competitiveness or attractiveness of their social environment? In songbirds, there are many components of birdsong that can vary in their ability to attract mates and to repel rivals. So these include things like repertoire size, or for example, the number of song types that an individual sings, song content, or the actual syllable types or song types contained in a repertoire, and performance-related traits. These are quantitative traits that are irrespective of song content. So things like song length, song rate, and amplitude. And these, the value of these performance-related traits can vary in space and time for a population. My project focuses specifically on a performance-related trait called trill performance. So before I tell you why I'm interested in trill performance, I want to introduce you to trill performance. So here I have a spectrogram of a Lincoln sparrow song, where we have time on the x-axis and frequency on the y-axis, and the darkness of these elements indicates their amplitude. We can break down the song into individual syllables, so these individual units of that we call syllables. And where you see the same syllable type repeat consecutively, we call that a trill, right? So this song has 6 trills. So trill performance is based on the relationship between trill rate, or the number of syllables per duration of the trill, and frequency bandwidth, or the range of frequencies that the syllables of that trill span. And this is an interesting relationship because there's evidence for a conserved trade-off between frequency bandwidth and trill rate. Podos, in his 1997 evolution paper, plotted frequency bandwidth against trill rate for multiple trills from multiple different sparrow species. And you'll notice this triangular distribution of points. If you plot a regression line through just the maximum frequency bandwidth trills, you see this negative relationship, indicating that as trill rate increases, the maximum possible frequency bandwidth for that trill decreases. So a good way to understand trill performance is through clapping. So I want everyone to clap with me. We're going to start by doing very small claps as quickly as we can. Okay, and then I want everyone to do much larger claps, but maintaining that same clapping rate. And what you may have noticed is that it's much more difficult to maintain this quick clapping rate when you're doing these large claps. We think the same thing is going on in the vocal tract of the songbird. So it's difficult to do these broad frequency sweeps at a fast unit time. So when we talk about trill performance, then high performance songs are those that maximize both frequency bandwidth and trill rate. So we have this, what we call upper bound regression line. Those trills would fall closer to this line. Low performance trills are going to be those ones that fall farther away from this upper bound regression line. In my study species, the Lincoln sparrow, we have evidence that females prefer high performance trills. So they prefer songs that contain high performance trills compared to songs that contain low performance trills. There are also a lot of studies that have measured the role of trill performance in male competition. These studies have found that generally the aggressive response to trill performance varies across species and different stimulus values. Kind of a pattern that comes out is that depending on the species and the stimulus value, males are either aggressive towards high performance trills or they appear to be intimidated by high performance trills. Another perspective of looking at the role of trill performance in male competition is to measure males' own modulation of trill performance. And so a study in swamp sparrows found that males elevate trill performance in aggressive compared to neutral context. In order to determine if trill performance is a competitive signal in Lincoln sparrows, I used a simulated territorial intrusion where I artificially manipulated songs to be of high or low trill performance and exposed these to territorial males. So before I get into the methodology of that experiment, I'll first show you how I do this experimental manipulation of trill performance. And so keep in mind that this is the same manipulation that I do for all of my experiments. So I'll show you this methodology now, but it's something that I use throughout the talk. Okay, so here we have two identical copies of the same song, and I'm going to focus just on this central trill. We can zoom in on this central trill, and I'm going to manipulate the top copy to be the high performance version by removing 15 milliseconds of silence from the intersyllabic space. And the bottom copy I'll make the low performance version by inserting that 15 milliseconds of silence. So I would repeat this same manipulation for all of the trills in all of my experimental songs. In my field simulated intrusion study, I constructed 10 playback file pairs. Each pair contained 3 to 5 songs from a single male. And here is just one of those songs, the low performance version and the high performance version. So that's the low performance version and the high. So I took my 10 playback file pairs to our field site in Mullis Pass, Colorado, where we study social behavior and breeding behavior in Lincoln sparrows. I randomly assigned 19 Lincoln sparrows to one of these 10 playback file pairs, and I placed the speaker in the male's territory and exposed him to the versions of these treatment songs over a 2-day period. And I alternated the order of treatments between males. So during this simulated intrusion, I observed the males' aggressive behaviors and I recorded their songs both during and after. Okay, so I observed several different aggressive behaviors, but aggression is a composite trait of all of these measurements of behavior. So I used the principal components analysis to get one measurement of aggressive behavior, where positive values indicate that males spent more time close to the speaker and had a shorter latency to approach. Negative values indicated that males spent more time far from the speaker and took longer to approach. And I found that males were more aggressive towards those playback songs when they were exposed to the high performance version compared to the exposure to the low performance version of those songs. Furthermore, I found that males did modulate their own trill performance in response to the intruder songs. Now, the pattern or the direction of results was a bit different than I would have predicted, but I found that males elevated their own trill performance in response to the low performance intruder compared to their response to the high performance intruder. Furthermore, this elevation in trill performance was actually related to male size. So following exposure to these low performance songs, it was larger males that tended to increase their own trill performance. Okay, so I think we have a decent story about the function of trill performance in Lincoln sparrows. I have some data and some evidence that trill performance potentially functions as an honest signal in this species. We know that female Lincoln sparrows prefer songs with high performance trills, suggesting that they may obtain some sort of benefit from mating with males that have high performance trills. Furthermore, I have evidence from the study I just discussed indicating that trill performance functions in male competition. So high performance trills are competitive signals for males. They respond more aggressively. And males modulate their own trill performance in competitive interactions. Furthermore, the positive relationship between the elevation in trill performance and male size suggests that males may be constrained in their ability to elevate trills, further suggesting this may be an honest signal of male competitive quality potentially. So that's it, right? Are we done? We can all go to lunch. Like everything in biology, there are always layers upon layers of complexity. When we looked at the songs that we've recorded from males at our study population, one thing we've observed is this annual variation in trill performance in the prevailing social environment. So this is the average and standard error of all of the trills from males that we've recorded during the breeding season at our field site. So suggesting that potentially from year to year, individuals are experiencing differences in average trill performance in the prevailing social environment. And I predicted that both males and females then would incorporate information about trill performance in the prevailing social environment and would modulate their social behaviors accordingly. And of course, these changes in social behavior due to the social environment will be mediated by underlying changes in the brain, also known as the big black box. So I wanted to open that box to begin to understand how the brain responds to these complex social signals. And there are multiple ways that the brain could integrate information about the social environment, so through perceptual regions, through motor output, and through motivational regions of the brain. I proposed that the social environment could affect behavior through experience-dependent changes in perception. So if the social environment influenced sensory pathways that process information about social signals, this could influence perception of those signals and behavioral responses to the signals. So the next question then is, where in the songbird brain are they processing these signals, the song signals? So if we look at, this is a sagittal or side view of the songbird brain. If we look at the auditory pathway, auditory information ascends from the hindbrain through the midbrain nucleus and thalamic nucleus up to the auditory telencephalon. And there are two regions of the auditory telencephalon, the cotomedial mesopallium, which I'll refer to as CMM, and the cotomedial nidopallium, which I'll refer to as NCM. And these are responsible for higher order processing of song. For example, these regions selectively respond to song quality. An example is that in female songbirds, exposure to more attractive songs leads to greater levels of gene expression in these regions compared to females exposed to less attractive songs. Catecholamines are a group of neural compounds that could mediate the changes in sensory structures in response to the social environment. Catecholamines act as neuromodulators by modulating the excitatory or inhibitory effects of other neurotransmitters. And so they function to integrate information about the internal state of the organism with information about the external environment. The catecholamines that I'm focusing on, you probably have heard of, are dopamine and norepinephrine, also referred to as noradrenaline. And the function of these catecholamines is conserved across vertebrates. In the central nervous system, it's been implicated in regulating attention, reward, and sensory processing, and learning to optimize behavior. If we examine dopamine and norepinephrine responses to song in CMM and NCM, one thing that we found, for example, is that there's an they're elevated in response to attractive song in, for example, female songbirds. In the auditory telencephalon, these catecholamines are responsive to song and to variation in conspecific song. And just a bit about the anatomy. I won't go into too much detail, but the catecholamines are synthesized in nuclei and cell bodies in the midbrain that send projections throughout the brain, including to the auditory telencephalon. And there's good evidence for high levels of both of these catecholamines in CMM and NCM, suggesting that there's some function in those regions. There are also synthesizing enzymes in CMM and NCM, as well as receptors in these regions. Okay, so I proposed that trill performance in the social environment could affect social behavior through modulation of catecholamines in the auditory forebrain or telencephalon. And in line with this proposed mechanism, I tested two hypotheses. I first tested the hypothesis that trill performance would affect preference behavior in catecholamines in females. And I next tested the hypothesis that trill performance during development would affect adult singing behavior in catecholamines in males. And I'll start with my first hypothesis. So as I've discussed somewhat, when evaluating a stimulus, individuals often assign its value relative to compared stimuli. And this phenomenon is known as the contrast effect, or can result in a contrast effect, which is illustrated here in the Ebbinghaus illusion, where the size of the center images are the same, but they appear relatively larger or smaller compared to the surrounding images. These contrasts facilitate decision-making by allowing individuals to evaluate one stimulus or one option in response to other possibilities. And so if we think about the decision-making process of mate choice, if average male quality varies temporally or spatially, as we've shown temporally for trill performance, this would vary the context in which females are making mate choice decisions. And females would benefit from evaluating a potential mate and adjusting preferences for that mate based on the quality of other males in the population. This could assure, for example, that mating still occurs even when high-quality males are rare or absent. My first question was, do females contrast the song attractiveness with the prevailing social environment? To ask this question, I used that same experimental manipulation of trill performance, and I manipulated 18 songs, 3 from each of 6 males. And here's a low performance version of a song, and the high performance version. Okay. So I exposed females to either the high or low performance versions for 7 days. And then on the 8th day, I exposed all females to the same novel songs that were of intermediate performance. Okay, before I go into the results, I just want to orient you to my results display. So here, I'm showing days 1 through 7 of exposure to the treatment songs. Here, I'm showing the response to those novel songs on day 8. And on the y-axis, I have phonotaxis time as a measurement of attraction to the songs. So on that first day, I found that females were more attracted to the treatment songs when they were exposed to the high performance versions of the songs than the low performance versions. And then I continued to expose the females to the same songs, played in random order, day after day after day. And probably as expected, by the 6th day, they had largely habituated to these same songs and were no longer showing a differential response. I then assessed their response to the novel songs, and I found that females that had been exposed to the low performance songs for 7 days were more attracted to this novel song of intermediate performance on the 8th day. So again, all females in this group were being exposed to the same exact songs. Their response to those songs was dependent on what they've been hearing for the past 7 days. And this finding suggests that females do contrast song attractiveness with the attractiveness of songs in the prevailing social environment. This finding raises the hypothesis that the social environment could affect sensory processing of song performance or of song. And if catecholamines are mediating the influence of the social environment on attraction to song, then I would expect exposure to songs of high or low trill performance would affect catecholamine levels. So my next question was, does experience with trill performance modulate catecholamines in the auditory telencephalon? A previous study did a few studies have investigated this question. The one I'll focus on is a study in European starlings that exposed females to 1 week of either less attractive short bout songs or more attractive long bout songs, and found that norepinephrine activity and dopamine activity were higher in the group that had been exposed to 1 week of long bout songs, suggesting, or at least raising the hypothesis that that catecholamines could mediate any influence of the social environment on behavior. But if catecholamines are mediating the effects of the social environment on behavior, I predicted that the effect of song on catecholamines should be detected not just at the end of 1 week of exposure, but much earlier in the exposure. So for this study, I was interested in looking at this early catecholaminergic response to these treatment songs. So I exposed females to just 5 hours of high or low performance songs, which would mimic 1 morning of exposure to songs, right? I dissected tissue from the CMM and NCM, and then used tissue extracts to measure catecholamines and metabolites using high pressure liquid chromatography with electrochemical detection. And I'm measuring the catecholamines, but when catecholamines are secreted from the cell, they can be broken down into their metabolites or byproducts. So we can measure metabolites as a very rough estimate of secretion, and therefore when we measure both of these, we can get an idea of catecholaminergic activity. I found that within NCM, both norepinephrine and its metabolite were differentially affected by the treatment songs, with an elevation this time in the group that was exposed to the low performance songs. So exposure to these low performance songs for 5 hours elevates both norepinephrine and its metabolite in the NCM compared to the group exposed to the high performance songs for the same amount of time, suggesting that both levels and secretion of norepinephrine are elevated in this low performance group. Okay, so I found that experience with trill performance does modulate catecholamines, specifically norepinephrine. What conclusions can we draw from this finding? So this does raise the hypothesis that potentially norepinephrine in NCM mediates the effects of the social environment on attraction to novel song that we observed in this contrast effect experiment. Of course, this is a hypothesis. I would actually need to manipulate norepinephrine in NCM to test this hypothesis. We do know a bit about the function of norepinephrine, even in NCM. So broadly, it modulates attention and focuses attention and enhances neural responses to salient stimuli. A recent study in zebrafinch found that application of norepinephrine directly to NCM enhanced the neural response of those neurons to song over kind of background noise or firing patterns of the neuron. So this does raise the hypothesis, could norepinephrine function to increase responsiveness to song in general in these low performance females? Again, I would have to actually go in and manipulate it to test that hypothesis. I found no effect of the treatments on dopamine.
Susan, before you go on, do you have any idea of what the no sound level of these catecholamines would be, so that you would know if it's that higher level in low performance is because it's increasing versus decreasing? Do you, from any of the studies?
Yeah, so I don't have a good sense of that, primarily because it's been difficult to include like a silent group in these studies, mostly because of sample size issues. But that is a really good point, that right, it could be elevated in the low performance females, it could also be decreased in the high performance females. Okay, so I've shown that females use trill performance in the social environment to adjust behavior, and that there's a corresponding change in norepinephrine and catecholamines in the auditory forebrain. But I thought, well, could developing males also use trill performance? And potentially, could these developing males use trill performance to adaptively invest in their own songs that could potentially benefit them in adulthood? I'm referring to specifically is what's known as socially cued developmental plasticity, where individuals use the competitiveness of the social environment to adaptively anticipate trait suitability for a later life stage. And this has been shown nicely in a study in field crickets where males were reared, so as juveniles they were reared in an environment that contained either no song or that contained an adult sexual song signal. So these males that were reared with song were essentially developing in an environment where they were exposed to a lot of reproductively active and competitive males compared to the males that were reared with no song. These males that were reared with no song were more likely to act as satellite males, so spending more time near a speaker, indicating that they're acting as satellite or sneaker males. They were also less likely to invest in body condition and residual or in reproductive tissue. So overall, if they're reared with less song, they're investing less in adult competitive reproductive tissue and behavior. Now, this finding could potentially happen in Lincoln sparrows, right? So Lincoln sparrows that are reared with high-performance songs might perceive or might invest more in their own songs and in their own competitive singing ability to adaptively anticipate potentially the social environment they would experience in the future. This is a little more complicated because we know that sparrows do learn their songs, so we could also argue that potentially they're just copying the trill performance of the tutor song without actually investing differently in their songs. But there's evidence from swamp sparrows that they don't necessarily accurately copy trill performance and trill rate. So in a study that studied the male's response to tutor songs that had been artificially slowed, they found that males elevated trill rates when they copied artificially slowed trills. In another study, they found that males decreased their trill rates when they learned from artificially elevated trills. And that's shown here in this swamp sparrow song. So trill performance is a performance-based trait. You have to potentially put some effort into the skill of having a high-performance song. There could be constraints in terms of genetic constraints or condition constraints. And so juveniles may need to differentially invest in high-performance trills in order to accurately copy those. My next question then was, does developmental exposure to trill performance affect adult song? And I went about answering this question by exposing males to, juvenile males to either 18 songs that I manipulated to be of high trill performance or the same 18 songs that I manipulated to be of low trill performance. I exposed them to these songs every day from age 38 post-hatch to 95 post-hatch. Now, we don't know exactly when Lincoln sparrows are learning their songs, but we know a bit about the schedule of song learning from closely related swamp sparrows and song sparrows. And from those studies in sparrows, there's evidence that within the first 100 days of life post-hatch, these sparrows are going through what's called the sensory phase. So they're not singing their songs yet, but they're memorizing tutor songs. They're sensitive to tutor songs. And after this stage, they're less likely to be able to memorize tutor songs. So my experimental exposure to the high and low trill performance songs fell within that period of the sensory phase that we know of in closely related species of sparrows. Following the exposure, I reared all of the males in a mixed treatment aviary to adulthood. And then when males were approximately 314 days post-hatch, I placed them in these sound isolation chambers and measured what I call baseline singing. So this is just the males singing in a silent enclosure. I then exposed all males to 3 days of novel songs that were of intermediate trill performance, and I call this playback singing. So there's a lucky enough to one day be, this was when the males were in kind of this mixed treatment group, but one of the males was just singing away in his cage. So I opened the door and stuck my phone in there. Should work. So this is an example of this crystallized song. But my question was, of course, how do these developmental treatment groups affect aspects of crystallized song? And I first wanted to ask, how do the developmental treatment groups affect the male's own trill performance? And so I tested the influence of the treatment and interaction with day across all of the days of song recordings, and I found that across all of the days there was in general an effective treatment where males that had been reared with these high performance treatments had higher performance trills themselves compared to males that had been reared with low performance treatments. And no findings in terms of a difference in slope across these days, which was one of my interests. Okay, so this finding indicates that developmental exposure to these higher low performance trills influences the male's own adult trill performance. And as you may recall, there were two, so trill performance is a composite trait, and there are two measurements that go into determining or calculating trill performance. One is frequency bandwidth, so I did examine if the developmental treatments influenced frequency bandwidth, and I found no effect. And the other was trill rate. So I also examined if the developmental treatments influenced a male's trill rate. And what I found was that over the days of exposure to, or the days of song recording, both the baseline and exposure to the playback, the males that had been reared with the high performance treatments had a higher or faster trill rate compared to the males that had been reared with the low performance treatment. And no real statistical difference in the slope across those days. Okay, so in answer to my first question, I found that developmental exposure to high compared to low performance songs increases a male's own trill performance and trill rate in adults. So what can we conclude from this finding? This suggests that males are learning in some aspect the trill performance and trill rate from their tutor songs. They are differing based on the exposure during development. And so this does raise the question, well, is trill performance an honest signal if a male can just learn the trill performance of the tutor song? Is it actually reflecting any aspect of male condition or genetic quality? One possibility is that it is, but that males are investing differently in trill performance based on the developmental social environment, potentially at the expense of some other unmeasured trait or aspect. And another thing that this brings up is that the trill performance from 1 year could contribute to the social environment in subsequent years. If males are copying the trill performance that they're learning as juveniles, their trill performance the next year could reflect what they've been exposed to previously. And finally, I wanted to know if catecholamines mediate the influence of this developmental social environment on adult song, potentially through mediating or changing perception of that tutor song. I would expect the trill performance during development to affect trill performance or affect catecholamines in developing and in potentially in adult males. So coming back to my methods outline, I first measured catecholamines in the CMM and NCM of males after just 1 week of exposure to these high or low performance songs. Males were approximately 47 days post-hatch. And I found that within this group, I found that exposure to 1 week of high compared to low performance songs elevated two dopamine metabolites, so a primary and secondary metabolite of dopamine in the NCM. So this indicates that 1 week of exposure to high compared to low performance songs potentially increases dopamine secretion and activity in the auditory telencephalon. Okay, so I'm finding that developmental exposure to high performance songs increases dopamine in the developing CMM. What can we conclude from this finding? Well, we know a bit about the function of dopamine in the central nervous system. There's evidence that it mediates learning and also regulates synaptic plasticity. And so one possibility is that the treatments could influence synaptic plasticity in the auditory telencephalon, and therefore influence the development of the auditory telencephalon and its perception of song. Dopamine is also implicated in regulating reward processing. So this raises the hypothesis that the treatments could actually differ in their salience or in some sort of reward value for these developing males. In which case I would predict that the high performance drills, for one reason or another, are of higher value for these developing males than the low performance treatments. Again, what I have are correlational patterns, and so these are hypotheses that would need to be tested by actually manipulating dopamine during development, specifically within CMM. And I found no influence, although I did measure norepinephrine, I found no influence. Okay, so then I also thought, well, potentially this developmental exposure to the high and low trill performance could have long-term effects on catecholaminergic activity in the auditory telencephalon if it's having long-term effects on some level of auditory perception or sensory processing. So I also measured catecholamines in these adult males after recording their songs for 4 days. And I found no effect on dopamine in adults, but I did find an effect on norepinephrine metabolite in adults. So in this case, the males that had been exposed to this low performance developmental environment way back in development had higher levels of norepinephrine metabolite in CMM compared to the males that had been exposed to these high performance environments in development. And so this finding indicates that developmental exposure to trill performance has long-term effects on this sensory processing region of the brain. Okay, so in summary from these points, I found that exposure to dopamine, or exposure to these developmental treatments increased dopamine in the developing CMM and decreased norepinephrine in the adult CMM. So what conclusions can we draw from this finding of a decrease in norepinephrine in the adult CMM? So as I mentioned earlier, norepinephrine is known to enhance neural responsiveness to sensory input. And there are two potential explanations for our finding of a decrease in norepinephrine in the CMM. One is that I measured norepinephrine after 3 days of exposure to these intermediate performance songs that had been novel to the males. So these songs could be more salient for the males that had been reared in the low performance environment and that were developing and singing these low performance songs themselves. So this increase in norepinephrine could reflect a response to the intermediate performance songs. A non-mutually exclusive hypothesis is that the developmental environment could have a long-lasting influence on norepinephrine levels. So potentially some baseline level of norepinephrine or noradrenergic tone could be elevated in males that had been exposed to these low-performance environments during development. While it's a bit harder to draw conclusions to this aspect of the study, it does indicate that at some level of auditory perception or the auditory telencephalon, these developmental treatments are having a long-term change in the brain. And there was no influence on dopamine in these adults. Okay, so what can I conclude from all of these studies and from this dissertation work that I presented. So we found that trill performance in the prevailing social environment varies annually, and I was interested in both the behavioral and neural response of males and females to this social signal that we know varies annually. And I found that both males and females exhibit behavioral and catecholaminergic sensitivity to trill performance in their social environment, raising the hypothesis that these catecholamines could actually mediate social environment dependent behavioral changes. However, I found no consistent pattern between the sexes or between the different ages in the effect of the social environment on catecholamines. So this isn't necessarily surprising if you think of the role of neuromodulators in integrating information about the internal state of the organism with the external social environment. We might predict that catecholamines would function differently in response to these different stimuli in different groups of organisms. But overall, I think that this work provides insight into the role of life experience on plasticity in the brain and in behavior, and also provides an ecologically relevant framework to investigate both these neural and behavioral responses to complex social signals. Okay, and that's it.